So let's begin with science. With the help from the Scientific Advisory Committee, we put some thinking into this program, and we have hopefully 2 attractive sessions for you. So in the first session, we start in the past and we go into the future. And we're very fortunate to have Dr. Swanson, Maurice, present history, progress, and the future in myotonic dystrophy. And then the second part, in the second presentation, we will hear from Dr. Bassez about the French registry and really learn and hopefully hear what a powerful and state-of-the-art registry can do for the purpose of drug development, for the purpose of informing public health, and also position, hopefully, for the future when we need to integrate genetics and genomics. And then we look far into the future and we talk about gene editing approaches, and this will be done by Antoine. And with this, Maurice, the floor is yours.
Thank you, Andy. So as Andy just said, my task was to describe what has happened in the past in terms of therapeutic strategies. I'm going to talk a little bit about background, so the fact that myotonic dystrophy is not alone as a disease. It's a set of many diseases now caused by short tandem repeat expansions, and then go a little bit into what has happened with myotonic in terms of therapy development, and then morph into 2 specific areas which I think have proceeded the fastest right now. And I've tried to create a presentation that's all anonymous. I won't be mentioning authors, including myself. So onwards, this is my disclosure. So short tandem repeat diseases are now a family of diseases started by FRAX-A and spinal muscular atrophy in the early 1990s when these repeat expansions were discovered. And one thing I always like about this slide, if you look at the distribution of these repeats, if you look at the 5' UTR, the 3' UTR, you notice that they're trinucleotides. And in fact, in the 3' UTR, they're just CTG trinucleotides. But you look at the promoter and the intron, it's a much more divergent set of sequences. Why? One possibility is that, obviously, in the 5' UTR, the coding sequence and the 3' UTR, they all play a role probably in protein production. So you're scratching your head and, wait a minute, 3' UTR, what's that doing? There are not only upstream morphs, but downstream morphs which control protein production. So that may be a rationale for why that exists. But there's much more latitude in introns and promoters in terms of what type of repeats can exist. So we're here to hear about myotonic dystrophy, which are, I'm sure I don't need to convince this audience, they're a fascinating set of diseases. DM1, myotonic dystrophy type 1, you can see the repeat levels in affected patients, 50 to 4,500, probably more than 4,500 repeats. Myotonic dystrophy type 2 in the CNBP gene, 75 all the way up to 11,000, and probably even exceeds 11,000. On a southern blot, all you see generally is a smear. So why is DM2 so unleashed in terms of repeat expansions is one of the central questions that I think the future has to answer. I don't need to present this to this audience really, but I just wanted to cover what are the therapeutic challenges of looking at this incredibly multisystemic disease. So in muscle, you have myotonia, of course, variable in DM2, as Giovanni will tell me later on. Muscle weakness in terms of muscle weakness and wasting and disabling pain in DM2. The heart, cardiac conduction abnormalities, less common in DM2. And the brain, excessive daytime sleepiness, common to both DM1 and DM2. And, for instance, learning issues in the congenital population. Cerebral atrophy in the older population. So current symptomatic treatments are very beneficial to the affected individuals. Devices, rehabilitation, surgery are all important ways in which you can intercede and impact this disease progression. But of course, drugs are tremendously important. Diabetes, insulin metformin, which is a complex 1 inhibitor and AMPK activator. The GLP-1 receptor agonists have also come online now. In terms of muscle pain and hypersomnia, you can read the list. So what about the therapeutic strategies, however, to block disease onset and progression? And that's why we're in part at this session. So what are the DM path mechanisms and how have those allowed the development of various therapeutic modalities. So let's start at the very top where DMPK is being transcribed bidirectionally, producing both the CUG repeat and the CAG repeat expansion. The sense strand, the CUG repeat, is more highly expressed compared to the antisense, but the antisense is expressed and upregulated when the expansion begins to expand more. The immediate impact of that is the CUG RNA is then packaged, as all RNAs are, into RNPs to be processed, hopefully out into the cytoplasm or retained in the nucleus in some cases. But in this case, the CUG repeat expansion results in the formation of what we termed RNA foci. And what are these RNA foci is an ongoing ongoing conversation about what actually these foci are composed of. Are they unitary single RNAs? Are they multimeric RNAs? There's some interesting work coming out about that right now. But anyway, the primary factors which are sequestered by these repeats, as of now, who knows what the future will bring, are the MBNL proteins. MBNL proteins then are deprived of their normal function in triggering a shift from fetal expression patterns to adult expression patterns. So it's really the bewildering aspect of this is that muscle-blind, RB-Fox, a few others, NOVA, are a set of adult factors which express adult isoforms. There are many RNA-binding proteins which are expressed in proliferating cells and during embryogenesis. So why is this balance so tilted towards proliferating cells as opposed to fully differentiated cells? Is an interesting question. Anyway, robbing MBNL, because it's sequestered in RNA foci, results in a problem with expressing adult isoforms in DM tissues, resulting eventually, for instance, in myotonia, as the example given here. But the other pathway is that as the repeats continue to expand, essentially all of the muscle-blind protein becomes titrated out of the free pool. And then you have naked RNA, which is susceptible to RNA export, and then once in the cytoplasm, susceptible to RAN translation, repeat-associated non-AUG translation. So that produces, in the case of DM1, homopolymeric proteins, and then on the flip side of all this mechanism is DM2. So not only is MBNL titrated in DM2, but so are the RB-Fox proteins. Now you're scratching your head and going, wait a minute, the consensus sequence for RB-Fox proteins is not a GC dinucleotide flanked by pyrimidines like it is in DM1 and MBNL proteins. But when you think about the sequence that RB-Fox binds, UGC AUG, UGCA is a GC dinucleotide flanked by a pyrimidine and a purine. MBNL3, for instance, also has a preferred binding site for that. So it makes sense RB-Fox binds and forms foci which are both RB-Fox abundant and MBNL abundant. Again, once you run out of those proteins, the naked RNA is exported and forms, in this case, tetrapeptide proteins. Both of these proteins, both the DM1 side and the DM2 side, produce proteins which are likely toxic to cells at high enough levels. What those levels are remains to be established. Again, an area of future research. So therapeutic testing in what do we use for models and try to move through all the way to clinic. So in IND studies, for instance, human fibroblasts and myoblasts have been extensively used, both DM1 and congenital myotonic dystrophy cells with large repeats. Primary cells are often used. They're often immortalized. And if you go down to the last bullet point, then what is the DMPK expression level and what's the methylation status of those cells is not always assessed. So I like to urge everyone to do that. In transdifferentiation, the nice thing about direct transdifferentiation is bingo, you switch a fibroblast into a myoblast, in this particular case by using an adenomyod. Notice the number of foci in those cells, which are now myoblast cells, compared to what on the iPSC cells on the left-hand side. The green is nestin and neurostem cell marker, And notice there are only a few foci in that particular case. So what's the difference here between direct transformation and the iPSC transformation? So the nice thing about iPSC cells, you can get isogenic controls, although coming from a past of yeast, it's not really isogenic. CRISPR does a bunch of other stuff. So you need a bunch of iPSC lines to make sure that all those iPSC lines are in fact recapitulating what you expect to be the truth. Co-culture allows you to look at cell interactions. 3D organoids, of course, are coming online, which have tremendous implications in the future. But then the question is, how is everything affected by epigenetic erasing and developmental level? As iPSC neurons don't make it much past P4 in a mouse. If you're talking about dopamine neurons, P0. So therapeutic testing on the animal side, what do we know? Constitutive, the expressing mice, which express CUG repeats, the HSA-LR and the DMXXL lines. You can see the misspicing is different between those two. Of course, the HSA-LR is skeletal muscle specific. DMXXL has expression in a variety of different tissues, so they both have their strengths. They've both been used extensively in therapeutic development. And then, of course, the conditional line shown on the left, which includes the EPA 960, GFP and DMPK 5 and 200 repeats in the TRED 960I repeat mice. All these have tremendous strengths depending on how you want to look at what question you want to answer and what kind of answers you want to come up with. So in terms of RNA foci variation, this is an excellent example of the differences. So you're looking at DM1 and DM2 on the right-hand side, CUG repeats and CCUG repeats in red, and the HSA-LR in the bottom. Notice it almost looks like grape clusters in the case of the HSA-LR, so very different than the presentation that you see in DM1 and DM2. So it's an important consideration to remember. Expansion lengths for disease, development and progression, and the roles of host gene, temporal and spatial gene expression. So that would be nice to have mouse models that recapitulated that. So that's been developed in DMPK expansion knock-in mice. So this, of course, using the strength of making repeats in vitro, because you can't make large repeats in cells, in bacteria and yeast, for instance, they get contracted. And then using rolling circle amplification, top right, to make lots of DNA that you can use as templates then. In this particular case, the largest template was 802. You wind up with the mouse, for instance, that have the correct integration event by CRISPR-Cas on the bottom left, and then eventually you wind up with mice that unfortunately don't have much of a phenotype, even though they're expressing repeats close to 1,000. How is this possible? They're being controlled by DMPK. So where those RNAs are being produced and how much RNA is produced clearly has an impact. However, the strength of this approach is that now you can uncover cells which are affected by those lower repeats. So basically, the first cells which are affected in this particular disease, perhaps, at least in a mouse model, we always have to make sure to caveat that way. But look at the RNA foci, somewhat reminiscent of the relatively small repeats, 250 to 210 repeats in the HSIR mouse. So what do we know about in terms of what the morphology of these repeats are and how they affect disease? That's another open question. DMPK mice which have up to 5,000 repeats, and you'll hear that later on this afternoon, in fact give a more accurate multisystemic DM1 phenocopy. So there is this threshold phenomenon. After you cross some threshold, which is north of 1,000 repeats, all of a sudden the mouse fully displays the phenotype, which I think has implications for what we also understand in human DM1 affecteds. Now, RNA-targeted strategies. So this is a table which shows RNA-targeted strategies, and this was written from the viewpoint of these strategies, therapeutics, which have been developed and actually made it to commercial viability in some cases. So antisense oligonucleotides are listed, ASOs, RNA interference, RNAi, U7 SNRPs and U1 SNRPs, trans-splicing, small molecules, RNA base editors, and engineered tRNAs. So notice each of these have their own strengths for instance, just the mechanisms. ASOs are both steric blockers and susceptible to RNA H cleavage if you make a gapmer, and we'll talk about that in a little bit while. RNAi has been a tremendous technology, especially in liver diseases up to this point. So you have both translational inhibition by microRNAs and slicing and turnover of the RNA by siRNAs. U7, again, like ASOs, can be used as a steric blocker, although there what you're doing is instead of having this artificial molecule, still an artificial molecule, but at least it's packaged in a biologically relative entity, that is a SNRP. So that has certain strengths that perhaps ASOs might not have. Transplicing is a technology still in the future, but has tremendous possibilities. Think of DMPK. If you can simply transplice out that last exon, so you replace the mutant exon with a wild-type exon, then you don't have to gene edit. You've done the editing at the RNA level. Small molecules, we'll talk about RNA base editors and engineered tRNAs, which will probably be a point of discussion in other talks. So I'm going to talk primarily about ASOs and small molecules because those are the ones that have progressed further in clinics. So this is just a cheat sheet now of a review written in 2025, and it should be somewhat concerning to this audience from a certain point of view. ASO-based trials, DMD, there are 12, 4 for DM, and FSHD, 2. So more trials are needed definitely in DM1. So let's move on to antisense oligonucleotides, really starting with Khurana's work in the 1950s, all the way going through current and modern strategies. We have a variety of ways of making antisense oligonucleotides more stable in the serum, better biodistribution by binding serum proteins such as human serum albumin and other factors. And those are all done by phosphate modifications such as phosphorothioate, sugar modifications, which includes 2-O-methyl, 2-methoxyethyl, 2-fluoro-RNA, not RNA, constrained ethyl, cyclic nucleic acids, and linked nucleic acids. So notice changing the phosphate, changing the sugar, or doing both are effective in the case of morpholino. So it's this diversity of ASOs and the kind of chemistry you can do on ASOs, which is its strength. And in fact, a lot of the new molecules that are being used at AS4, it's a mixture of different kinds of ASOs that are being used in the same ASO. So ultimately, this winds up with designs for the RNA. The most important on the bottom in the blue is nusinersen. So this is the A which is used to block an intronic splicing silencer downstream of spinal muscular atrophy, spinal motor neuron gene, SMN, exon 7. So this blocks the splicing silencer and allows inclusion of SMN2 to remediate spinal muscular atrophy, and it's been a tremendously effective therapy. Up from that, and below that, I should say, first of all, the siRNA design, similar kind of strategy, of course, 3' overhangs, and then mer design where you have a DNA flanked by RNA sequences where those RNA sequences are modified by the structures I'm just showing you above. So, history. So this is history, and it's not meant to boggle your mind, but it is an interesting history. So I start off with a RSV, by the way, is not respiratory syncytial virus. In 1978, it was Ralph sarcoma virus. It's a very interesting experiment. It was done, that's reference number 1 down on the bottom left, where people took a 13-mer, realized that the repeated sequence on 35S Ralph sarcoma virus, 35S RNA, and made a 13-mer which bound both the 5' and 3' end that inhibit viral replication and also translation. So as far as I know, that's one of the first demonstrations that antisense technology could be effective. Then move on to MBNL and splicing blockers later on, much later on in 2003. This is a retrovirus done by people in this room where it contains a CUG13 and also a 3' UTR sequence. This is just an antisense RNA, which blocks DMPK, which binds to DMPK RNA and basically blocks the association of MBNL proteins. So that drops DMPK RNA levels. And that was the first demonstration that I know of that, in fact, perhaps blocking interactions of that RBP in the RNA sent the RNA down a decay pathway. In 2007, there was an attempt to actually cure myotonia by using a ASO which targeted the 3' splice site of CLCN1 exon 7A. So that worked, and in fact blocked E7A splicing, that cryptic exon, which sent the RNA down the nonsense-mediated K pathway, and so it was a cure for myotonia. But of course, there were several problems with that, and that is that myotonia is not the only outcome affected by alternative splicing. 2009, a CAG morpholino was used, CAG25 was used to block MBNL interactions, and a CAG7 ASO was essentially did the same thing in DM myoblasts. So most of these tests were done both in cells and in animals. So that was a tremendous strength to see it both in cellular and in vivo. 2012, gapmers came online. So you have both LNA and MOE CAGs, EPA 960. So this is something that's targeting just the repeat. So this would be allele specific, not non-allele specific. In other words, it's hitting just the mutant allele as opposed to both the wild type and mutant allele. So that definitely has potential strengths. That was followed very quickly by an MOE which targeted DMPK. And again, these are gapmers, so they're basically degrading the RNA. And in this case, in the latter case, you're degrading both the wild type and the mutant allele. Following that was a constrained ethyl gapmer, which eventually made it into clinical trials, unfortunately failed clinical trials, not because it didn't work for everyone, but because it didn't work enough in terms of the population distribution. That was followed by antagomers or anti-mirs. So one issue might be that if you raise the level of MBNL proteins, what might happen is you overcome all of those splicing defects. And that seemed to be true, that in fact, the ASOs targeting 2 mirs, which regulate the level of MBNL1 in this case, in fact, reduce missplicing, increased MBNL levels, so fairly effective potential therapeutic. 2019, the introduction of cell-permeable peptides, that's CPP, PMO blockers. So this is a molecule which binds directly to the CUG repeat, exactly what we heard a little bit about before, and targets only the mutant allele. 2023, the CPP PMOs were incorporated into the anti-mirs, and in 2023 also, it's a really interesting study where there's a comparison between a CUG expansion blocker and a DMPK degrader. So just ASOs versus gapmers. And the winner, according to this paper, and we can discuss it at the bar, is that, in fact, blockers were more effective. They both dropped DMPK levels and reduced RNA foci. The blockers reduced missplicing and reduced off targets better than the gapmer. Finally, in 2023 was an active year, you had a U7, which had been designed to have different SM binding sites and optimized so that its 5' end would now bind directly to mutant RNA. And that also reduced foci and missplicing. So that's an antisense U7 siRNA technology. Small molecules. So this is another interesting, a little bit like the early work with RSV that I pulled out in 2024. So this was a study that was done in kidney cells, which had 160 repeats, and they used genotoxic reagents. All of you are drinking coffee, right? Caffeine actually increased repeat expansions. The caveat here, don't worry, the concentrations were massive, so much higher than in your cup of joe. And aspirin reduced expansions. So caffeine, maybe because of its effect on ATM, and aspirin because it actually is a proliferative sensitive drug. So then we go into CUG blockers. You can see, I love chemical structures. Sorry, it's the chemist in me, so all these chemical structures are written up here. It started off with Hoechst dye, which has been known to be non-toxic and actually binds fairly well to CUG repeats. That's the 2009. That led into the discovery of pentamidine and neomycin B, which also bind those repeats. pentamidine had the strength that it was also already used and approved by the FDA for parasitic infections. So it could be used in humans without horrible aspects coming up. Although if you have a parasitic infection, you're not going to be taking it chronically, you're going to be taking it acutely. So they were effective because they, again, dropped the interaction of MBNL and CUG repeats and dropped missplicing. Another interesting molecule with triaminotriazine and acridine conjugates. Every time acridine comes up, I worry. But the issue here was very interesting, is that the acridine, of course, would intercalate in the double-stranded structure that we believe exists with CUG repeats. And the triaminotriazine would interact with the bulged U residues in DMPK. So it was an interesting strategy, and it also worked, drop missplicing in cells in mice. Bivalent ligands came in after that in 2013, followed by substituted naphthalene in late 2013, and then development, which is progression from the 2009 discovery of pentamidine to furamidine, which also had an effect of blocking MBNL CUG interactions and remediating splicing. That then moved into another era of 2015 also of looking at actinomycin D. Actinomycin D has the interesting property of binding GC-rich sequences. So if you can bind actinomycin D and inhibit Pol2 from getting across those repeats, you might have abortive transcription and turnover specifically of the mutant allele transcript. The concentrations that we all use to inhibit transcription in our cells, these concentrations are much, much less. So it is a viable drug, perhaps. My favorite name, CUG degrader, is CUGamycin or cougamycin, which was developed to both using HSALR mice and also cell models. Again, CUG expansions dropped and myotonia dropped. So this is a combination of a molecule which recognized the mutant RNA and bleomycin, which is known to cleave both RNA and DNA. So you had a binder and a clipper in one molecule. And then one compound, and this is where I think there's a major deficiency in the field. So everything I've talked about practically is in DM1, myotonic dystrophy type 1. So if you're sitting there and you have a family affected by DM2, you're going, wait a minute, where are all the DM2 therapeutics? This is one possibility. It's known that in DM2, in CNBP, the CCTG repeat is in an intron. The expansion of that repeat causes retention of that intron. That has several problems. One is that it can be exported into cytoplasm and be translated into those RAN translation proteins I talked about before. So wait a minute, if you could find a compound that actually CUG intron 1 retention, then maybe you would increase the CCUG turnover, and that worked. So just one very quick surprise is that, again, with small molecules, and we might talk about this, I think Darren may talk about this a little bit later, was discovered in CAG repeats for Huntington's disease. And this has a really interesting property, is that there are two small molecules. Risdiplam is now on the market for treatment of SMA. It has been in the market for several years and certainly been in clinical trials for a number of years. No obvious problems with the drug, massive problems with the drug. Both drugs also decrease PMS1, which is part of the mut-L beta. And I think Darren might talk about this, so I'm running out of time, so I won't talk about this. But the interesting thing is now you have a drug that actually can cause turnover of PMS1 and result in contractions. In this case, CAG contractions, but if you flip that, it's CTG, CAG contractions. So another possibility, but what I wanted to do is really introduce that from the concept of we haven't looked at the whole molecule space. We're looking at we're focused on a particular aspect of the pathogenic pathway, and that's why I tried to bring it up initially. So anyway, the overview then, I'll just go through this quickly: allele nonspecific, allele specific, and what I haven't talked is about transcription blocks, but those are not close to clinic right now. So additional therapeutic initiatives, I haven't talked about genome editing. We'll hear about that very quickly. Transcriptional modulation, which I think is another very important aspect of what could be in the future. And that's transcriptional modulation of all the characters involved in DM1 and DM2 pathogenesis. What we don't know much about is RAND translation and what's the impact of RAND translation in this disease. Is there an impact or not? I would say in the brain, almost certainly there's an impact. It's less clear in skeletal muscle and other peripheral tissues. So future research will have to deal with that. And then finally, very late are the proteins, PPR, pentatrichopeptide proteins, and PUF proteins, which instead of ASOs can be programmed to recognize very discrete sequences on RNA. So they can be programmed to bind CUG repeats. So this is a protein, PUF proteins, human proteins, which may not raise too much of an immune response. These are all the therapies in development from Andy, the other Andy. And then this is the approved RNA targeting therapeutics as of 2024. Notice all the DMD, 2 SMA, 1 ALS, no DM1. So DM1, DM2 therapeutic approval is the big question for our audience. Thank you very much.
Questions?
I just have one question. So DM1 or DM2 affects various tissues. Do you think we will find one treatment that will fix all? Or do we think we need to have very specifics, targeting skeletal muscle, targeting the CNS, targeting the heart?
Great question. And I don't think we know right now. We know certainly in animal models. We don't know in humans. I think that CNS is one issue. So how much of a particular drug gets into the CNS, if it's delivered IV, for instance, the concentration, is it? Because, of course, it's a sliding scale, too, depending on the individual that you've injected. What are their repeat sizes? What are the repeat sizes in the frontal cortex versus the hippocampus? It could be huge. And even within the hippocampus, what are the repeat sizes between individual hippocampal neurons? And the same sort of question about skeletal muscle you have, which are multinucleated. How many nuclei do you have to hit to get full remediation or even partial remediation of myotonia and block to muscle wasting and weakness?
Thank you.
Hi there. So I just wanted to ask, I noticed that ribotax were not up there, ribonuclease targeting chimeras, and I just was curious as to what your opinions are on that particular therapeutic or that strategy to degrade the CUG RNA.
So I'm not sure I heard the whole question, but it was about ribotax and how
Yes.
effective they will be at therapies.
Yes.
I think potentially very effective, but again, a more complicated strategy, a little bit like the cougamycin I talked about, right? Rather than a certain kind of pure chemical drug, ribotax have even more potential, but also a lot of therapeutic development has to be done to deal with that issue.
Thank you so much for great presentation. This was awesome. I have two questions. One is, I'm thinking about if you can comment on the amount of repeats you have in different cells could be quite different. If you have 50 CUG repeats, or if you have 4,000 CUG repeats, the amount of compounds you would need in a cell will be quite dramatically different. That's one question. And the other is, I wonder if the term allele specific could be, if we can use CUG specific instead, because there are like 70 something transcripts that have CUG repeats. So likely these drugs will affect some of these other transcripts.
Yeah, so I attack the first question first. I think that's an open question. That's the issue with just, as you said, for the variable number of repeats in individual cells, and especially between cells within a tissue population. I don't think we know at this point, and I'm sure that all of the individuals who are involved in therapy development in the room, this is an issue they tackle all the time. There's going to be tremendous variability, I assume, in the patient population in terms of the amount of drug, how frequently the drug has to be administered. It won't be, I think to a certain extent that's also true in SMA for risdiplam, for instance, but I'm not sure about that. The advantage of small molecules, easy to modulate the dose pretty readily. That's also true of ASOs. More permanent therapies, for instance, AAV therapies, less. We've been tackling this question. So again, it's the multi-systemic presentation. It may be that you have full remediation in a particular tissue and not full remediation in another tissue. So the first thing that comes to mind is skeletal muscle, heart muscle versus the brain. Do you have less entry of that particular molecule in the brain? And is it less effective in the brain? Because maybe the repeats are even more massive than they are. And this is definitely a problem with DM2, where the repeats are already quite large and the variability there is quite large. Great questions. I don't have answers, sorry.
Great. Thank you very much again. So next we hear from Guillaume.
Hello, everyone, and thanks to Andy, to Tanya, and Nadine, and all the MDF community for inviting. MDF is doing a fantastic job. So you can see, and when I received the title, which is a little bit sophisticated by Andy, it took quite 2 Zoom meeting to really make what would be the recontent, but finally, this is quite simple. This is what can we learn from the DM patient data? That would be, and I will a little bit cover the past, the present, and some future. But just to start for those that, because we are European with not that many today, and we do see that some of us, they contact separately, each one of us, while we are in the same center, the same institute, the same building. You have yesterday seen some use of immortalized myoblast cell lines, the DMSXL, and now there is a very exciting new inducible model that is in development in our lab. And I'm in the last piece in charge of this translational research. And there, I should add some MR and physio facility with the outcome measure. And of course, all this team that are here, which is led by Denis Felling, we have Geneviève, Mario is also there, is working together. So it's very exciting environment. And for those who are young DM scientists and would like to enter the DM field. So you had the presentation from Mario to enter for regarding a position to enter DM1. Coming to the summary, what I will present to you today is a little bit going to the past. What was the rationale for having a data collection for DM? What are some contributions to the field of this DM scope registry? And how currently we use, because we are in a particular very specific time with quite many clinical trials at the same time. So we need a powerful tool and infrastructure to recruit patients, to be efficient in that. And I represent some of the extension, European extension and international collaboration extension. And finally, be prepared for also the future, for future years and next steps. And I will briefly present, and maybe we'll discuss some of the next development that are needed for the DM community. Just have a look at the rationale. So it's complex and simple. What it is complex, it's because as compared to other neuromuscular disease, genetic, we're used with congenital myopathy or so on, they are purely muscle, and we can do good data collection and registry with good measure. Of course, due to the multi-system involvement and multi-organ involvement in DM, this is much more complex. So this is one factor of complexity. A second one is temporal, because DM patients, they are splitting from the birth to 80. So this is 2 axes of, and when they are combined, this is probably, of course, the most complex disease we are dealing with. Another aspect of complexity is the high prevalence for rare disease. And we will see that might be an advantage, but this is also a little bit of a challenge. So putting together the needs, the unmet need was and is still to improve the knowledge on DM1 for the community. There are tools like different data collection systems, but for some of them, they might not be complete or that with such a quality of the data regarding the mutation, for example, CTG repeat length. Do we have that for all registered participants? And might be incomplete. So this was the rationale to create DMScope, which has the objective of increasing the knowledge for all of us, for the DM community, but also to be efficient in, it was one of our first goals at the time regarding trial readiness and be prepared for trial patient recruitment efficiently, but also much more. So this is here. So these are the objectives, and I will, of course, so we are here currently, how to promote and accelerate the translational research for DM. And this is how we integrate it in the Institute of Myologie with really the basic research and clinical trial units. And the final aim would be, of course, to inform and to implement recommendation for the regulators, institution payers, and being prepared for the post-approval period. So I briefly summarized and took some of the, let's say, very few examples in the GM community. So altogether, you have two different axes. So whenever you have data collection with the use of data from patients, first one, you would like to have the more and more patient, the most patient you could collect. So this is one axis. The second one is that you would be interested to have a large data set, so many data of the patients. Ideally, we would be like here, having the entire DM population and all the data from the DM population. So of course, this is ideal and it's not real. So I give you a few examples of the reality, and I took some US. So first example could be the NDM study is really a natural history study. So you have a very large collection of data for limited sample size. Another one could be the MDF registry. And then you have people from, of course, the US, but also abroad. So you have many patients from different countries. This might be limited by the data you have from the patient themselves. So meaning that you have not all the data, and there are questions regarding what would be the completeness of the data quality. And we are a little bit in between with the DM scope. Want it to be restricted. This is national population. This is one country. It is a limited territory, quite a good size, and a quite, what we think, relevant number of data. So how we developed this, and we started from the Paris region at that time. So in a single center, just as a proof of concept, and progressively over the years, this extended nationally to all the region, north, south, east, west. And you have now, so it started with the adults, the M1 population. This is where I'm, and I'm more adult neurologist. And some years after, we extended to the pediatric. And now, nicely, we have the whole population from the congenital to the late onset adult population. This is quite important for epidemiologists not doing bias, so not biasing your population. And sometimes I had to question, why is DM scope quite efficient? And it's not because of me, I'm just the scientific coordinator, try to be active, but just that. So it's because of two factors. The first one I should mention here is the funding. And this is highly supported by a DM patient foundation, which is in our country, AFM Teleton. So now for more than 15 years, they are funding the registry, which has now a hybrid funding with a private partner and industry collaboration. And second, this is government, and sometimes government can help. And here with the rare disease plan, we had all these centers there where it was mandatory for them to participate if they want to be labeled as reference neuromuscular center. So it was two incentives finally that make it quite a rarity. And over the year, what we see is that gradually the number of included and recruited patients increased. And we just have reached very recently more than 4,000. This is very big, but now what we want is that to increase this curve, is that the patient we know for several times, several visits, we know when, not just once. And it's still possible because some of the patients, they say, I want to be registered, I want to be in. And of course, this is from patients association. We have to do that. And interestingly, you see this is the annual activity. And you see that now this really completely flipped. And now there is the annual report are much in favor or not the new patients. So they are not novel inclusion. Then this is how deep is the data collection. So it is quite deep in the DM scope. You have here all the data. And of course, not all the files do register all the data in all the centers. So this is here a combination we have to do, and briefly to separate the mandatory, the highly recommended, the optional one. And we can follow what is missing, what is there. And here you have two examples of the pediatric centers on the left and the adult centers. And we know what are the participants, what are in black the deceased participants, and which one are followed up at when. So if we want updated data and recent data. So like every project, it is separated in work packages. I will really focus on the operational one and what it can deliver. We just to let you know that this took years or a decade just to be there. So sometimes it's very exciting to have the data there, but just to remind that it took so time and resource. So what are some examples of contribution to the field? So first is that to really put all the patients we have all together and to try to stratify what could be DM stratification. So this is here. We started with 3 different groups, 4 different groups, 5 different groups that are here. They are all statistically different. So just to mention here that it is also very important that when you have a non-biased DM population, just to remember how this percentage, how the childhood form is elevated. Most of you or most of the people, they are thinking of the congenital. And when you say non-congenital DM1, this is all of this. And this is more than 95% of the DM1 population. So to me, non-congenital is a little bit non-informative. So it's non-something. But you don't know what are the patients. So you have to be more precise. And this is the stratification for them. Why? Because all of this subgroup, at least from the genetic level, they differ significantly. This is at the group level. And of course, the CTG size cannot be used individually to predict what could be the phenotype, what would be the clinical characteristic of a patient. These are the characteristics of the patient. When we plot this, and that we cannot read it, or it would take more time, but just to inform how rich it is. And we have separated some of the data that do not behave the same regarding different subgroups. So this is interesting. So I will not have time to comment here, but you have subgroup where it is decreasing with the form or increasing. Some of them are in stable and some of them are bell-shaped. So it's very interesting to know the behavior of this subpopulation. So there is no DM1. There are some specific individual across this group. And just to let you know that we recently had the ENMC workshop, which came to the conclusion that we have a consensus regarding literature and the designation. This is childhood onset DM1. So you have 4 groups, the congenital, childhood onset, adult, late onset. This is summarized here. So this is from this year, 2025. So please, any other terminology like the early adults or, I don't know, teenage or non-congenital or would be something juvenile, infantile, even we change our designation. And the good thing is that the NDM1 study already used for years the appropriate nomenclature. So this is very simple. We are talking about SMA. SMA had this classification in the 90s. So we have waited quite a long time. I started from the disease onset and birth. This is a question we have addressed more recently, is the survival. Very also briefly, we have studied what would be during the last decade, the survival of more than 1,400 DM individuals. And we see that during that period, 14% of them deceased. This is here the presentation of the median age of death. And this is 58. I know you can comment more on that, saying this is not that high. One of the explanations is that it is a non-biased population. In the very old literature, I just realized that it was focusing on the adult onset DM1. So this was neglecting the childhood onset, which are much earlier, more severe. This is the ratio of deaths during the year. This is here how it is. So you have the number, and it is slightly better survival rate in female compared to male, while being not significant statistically. You have here the question, what is the cause of death? And some of you may think, okay, cardiac involvement might be the first cause of death. This is not true or not anymore. Maybe because of the improvement in we have a very interesting session yesterday. And over the years, the cardiac management with the cardiac device, pacemaker, defibrillators really improved. So that might be an explanation for the decrease here in the cardiac deaths, while the first cause is respiratory in that disease. And there are other causes like cancer or other disease infections. So quite interesting. Of course, we all want to see what is the behavior regarding the CTG. There is a kind of weak correlation with CTG repeats and their survival. It cannot be used individually. More importantly, I do present here some of the contributor to that. One of them is the muscle impairment. And what is that? Read the split is from the MIRS 1 to 3 and between 3 and 4, 5. So here there are nearly two subgroups, one very favorable group with no or little or distal muscle involvement as compared to the MIRS 4 and 5, which is expanding to the limb girdle and the proximal involvement. So this is an indication and signal for lower prognostic regarding death. So this is the summary, and I covered here. Just to remember that the early onset disease is contributing to death as well as MIRS, and as well as BMI. So body mass index is contributing. So it was one of our, not a surprise, but here, just to see that really overweight, obesity can really have a negative impact on several aspects of the disease in DM1, as well as the respiratory involvement. Just to go to where we are now, what is the use of the registry? So I've given one example. So we have many clinical trials, and you have criteria. So we put this criteria in should start with here. So let's say you want people from a particular age range with specific muscle impairments, CTG size more than 150 or other parameters, and then pacemaker, yes or no, and other facts. So it gives the eligible population. And since Greater Paris is 10 million inhabitants, I'm very happy to recruit all the patients locally. And I have many questions, say, oh, would you not be interested by this family or this that are living? I say, we don't know anything about them. So why having this risk of screen failure or increase the patient burden? That would be the case. So what are the advantages today of having a registry? So first is that this is targeting the eligible patient, and it covers the major inclusion-exclusion criteria. Not all, I would say, and I have still the surprise of biological value. So I would really here emphasize, be aware, be careful of biological value in DM patient. We get some surprise. So this is something we do not control with the registry, but it is really identifying a kind of pre-screened population. This is very interesting. And the patient, they love this. They are close to the site. So it is, of course, reducing the cost, traveling, the burden of patients. So participants are the priorities. So think about them. And we have quite a low screening failure due to that. Managing AEs is also very facilitated. And in the future, that will align the preclinical trial data to the clinical trial and to the post-approval. So that will also benefit to the community. This is what we would like to be really a continuous data collection for DM patient. So briefly, I will present some of the international extension. So one of them is European one. The other one is transatlantic. So let's start with the North America, where we are. And so for years, we still have close collaboration with our Canadian colleagues for years, but we just realized that while our registry were very close, they were not similar. So why not? So then we go to this full harmonization. And sometimes due to the high prevalence in Quebec, I'm a little bit jealous, say, oh, we need more than 25 centers in France, while they do just have 5. So it's unequal. That could be, but it's good for their management. And this is 5 centers, 26. So this is life. More than the high prevalence, this is also a good example of synergizing from what is our respective domain of expertise. I'm more in the registries and the data collection, while Cynthia is, of course, an expert in the outcome measure assessment, and Hans Lockmüller in Ottawa more in the biomarker field. So we think that altogether this is a good network and it is a good consortium. But of course, it has regulatory, it has all these pieces of work that really are, we cannot see during a very short presentation like this. You've heard from Mario, very good development in Europe. This is sometimes the case. This is also a bureaucratic approach, and we contribute as DM Scope to some of the consortium to prepare what could be a European neuromuscular registry, which now started in 2023. And sometimes we make things complex in different countries and in Europe. That will not be the case for entry DM. So really, this will be nice for all the PhD students. So it is very briefly how it is. So for all neuromuscular disease, the very rare, the ultra rare disease that do not have registry, the clinicians, they can already put the data in the major registry. While we had some registry like the Duchenne data platform, SMA with SmartCat, DM with DM Scope. And we didn't want to recreate a new registry. So we create a hub where this can be interoperable and interconnected, not to duplicate and not to redo the data entry. So this is the model, very briefly. So this is where we are now, and with some contribution of the DM Scope registry to refining the DM classification, contributing to DM translational research. The last piece of work was this harmonization, and we had the ENMC workshop regarding the data collection and registry harmonization. This is very encouraging. Altogether, these are the strengths and the weaknesses of the registry currently. And you can see that having a large cohort is encompassing the two. So why? So it's good for a rare disease like myotonic dystrophy to have large cohort, but it is time-consuming, resource-consuming, and it's on both sides. So we are happy having some data quality, long-term longitudinal, linkage to biobank and to other tools. I will present very briefly 2 examples of that. Why we would like to have now is, of course, to reduce the missing data amount, and there is, of course, with hundreds of participants, and increasing and including PROs. So we do not have, this is really clinician expert centers based data entry. So this is very clinician oriented, and we would like to have the patient voice here. But of course, this is different system. The clinician, they access all the data. We would like the patient access some of the data. It's a little bit more complex, but this is future development. And the resourcing, I present some words. So we were purely financed by Patient Foundation, and now there's partnering with more and more companies. Just 2 words on what would be the future developments. It is now, DM Scope is not just as a whole, it's not a separate piece in a silo, and I want to get some linkage. So linkage to what? So 2 examples. We started, this is a study where I was thinking about the patient voice, and there has been a good survey, which is called the ENSA. So this is the DM population survey. And here we want to link what the patient think they have, what is their perception of the disease, to what they really have. And what they have is by clinicians, by experts of DM. And we want to compare this. Is it aligned? Is it separated? So this is quite interesting. I do not have present here, this is with Lupin Pharmaceutical, another aspect where we linked in the time DM Scope. So we have the retrospective data from the registry, and this will be bridged to a prospective natural history study. So we will have the 2 words combined all together. And finally, the last example of linkage is with the complete French population. So very briefly, this is the National Data Health System, which is called SNDS. And in France, because there is social security, 99% of the French individual, they are collected in that database. So these are more than 65 millions of people. So it's not proportional to the circle. And in that, and thanks to the registry, because you cannot find rare disease, there is no diagnosis. So we have aligned, and still anonymously, all the patients, and we are at more than 90%, which is very good linkage. So we know what have the clinician, and we know what are the expenses, what is the cost, what is the hospitalization rate, and we are working on that. So aligning, I would say, all of the data for these hundreds of DM participants. So this is quite exciting. Where we would like to go there, and this is good because it would be acknowledgements, even with the 5 minutes less probably, or I don't know. It is to be, so I think the next challenge is beyond, of course, France or France plus Canada, is to be efficient altogether as a community. So as an international data collection initiative. So for this, it was first, there are more than 20 registries over the world. They're all very good, but they are different. So the fact is really to get this harmonization. So we harmonize the data sets. So now, plus the classification, we will provide to the DM community this harmonized data set that is open and free and available to everyone. There will be the use of, we are in the clinical trials, but think about the next step. And the next step will be, hopefully, some of this drug will be approved, we hope. And there will come the question of the reimbursement and payers. So to get some real-world evidence from all of the data. We're in the process of being labeled by the National French Authority, Health Authority, which is called HS, to be the DM registry. Whenever there is the labeling, this is mandatory that all treated patients in France will go through that registry. That would be very nice. This is already the case for Pompe. For some of the very few, it's only one neuromuscular disease. So it's challenging, but we apply to get the quality and to get approved by this authority. And of course, what would be the benefit for all this linkage and having all this infrastructure in hands? It is really to increase and to inform the community. So to have longitudinal data of future treated patients, having real-world data, real-world evidence beyond the clinical trials, which are restricted to small population, to a selected DM population, and to support pharmacoepidemiological and economic analysis. This is, of course, not a single initiative. You will see how many people are there. This is the French coordinator team, and this is the team in Canada, but altogether, these are the number of contributors, and there are many, and thanks also to the company that are joining now. Thank you.
The last session of this morning, the last presentation, is looking into the future and gene editing. Antoine, take it away.
So hello, everyone. So first, I'd like to thank the organizer for inviting me speaking today. That's a real pleasure to be there. So so my name's Antoine. I work with Professor Vincent at Cardiff University and with the UK Dementia Research Institute. And so today I'll be presenting you a gene correction approach to treat expanded repeat disorders. So there's a lot of different expanded repeat disorders, which can be induced by different DNA repetitive sequence. I won't be into details for all of them. I'm sure like pretty much all of them, but today I'll be a bit more focused on repeat disorders induced by CAG repeats. So there's about 15 repeat disorders induced by CAG repeats. As of today, there's only one that has a potential cure, which is a Fuchs endothelial corneal dystrophy. The other 14 are still incurable as of today. In the lab, we are a bit more focused on Huntington disease, which is induced by CAG repeats in the Huntington gene. And the expansion is going to lead to production of a mutant Huntington protein that is going to form aggregates, and which is eventually going to interfere with a lot of gene expression and is going to lead to death of medium spiny neurons in the striatum. And there's a lot of different labs and companies trying to find a cure for this disease. We've seen some siRNA, we've been talk about ASOs, but we've also been briefly mentioning gene editing. And so for this, we have to reintroduce really quickly my best friend, CRISPR-Cas9. So CRISPR-Cas9 mainly is formed of two components. You have the Cas9 protein in light blue, and you have the guide RNA, which is in red. So what does Cas9 do? Cas9 has, I'd say, briefly, two main functions. The first one is to find what we call a protospacer adjacent motif or PAM sequence on the DNA, and it will bind to it. Once it's done, Cas9 can unwind the DNA. And then the second action that Cas9 does is it has two nuclease domains, one nuclease domain that can cut the targeted strand, and another nuclease domain that can cut the untargeted strand. And so it can lead to a double strand break. The guide RNA is basically going to show to Cas9 where it has to cut by interacting by base complementarity with the target DNA. So if I summarize the whole action, Cas9 tries to find the PAM sequence on the DNA. It binds to it, unwind the DNA. Then the guide is going to interact by base complementarity with the target DNA. And if all of this happen, then Cas9 can induce a double strand break. And so this approach has been used and tested for expanded repeat disorders. Doing this essentially, here you have Cas9 with two different guides. So one guide targeting here and one guide targeting here. And then what you end up with is you end up with a double strand break on both of the flanking sequence of the CAG repeats, which are in dark blue. And then doing this, you excise completely the repeats. And so you actually do revert the phenotype by excising the repeats. But there's a couple of drawbacks. The first one is using a nuclease that is going to induce double strand breaks, and that's error-prone repair. You're going to insert indels. The second issue is a high potential for off-targets. So Cas9 can sometimes cut at an unwanted place. And if you use a nuclease, then you're going to induce an indels at this place as well, which can be quite used. It's not allele specific, so you're going to excise the CAG repeats which are expanded, but also the CAG repeats which are not expanded. And in the best scenario, you would like to be allele specific and only target the expanded one. And lastly, is disease specific, because the flanking sequence for Huntington disease is going to be different for spinocerebellar ataxia or Fuchs or any of the other repeat disorders. And so seeing this, we're thinking Cas9 still seems to be an interesting tool. We might still be able to modify the approach to find something that might fix these issues. And so for this, we decided to try a nuclease. And so we use the same Cas9, but there is one point mutation in one of the nuclease domain that inactivate it. And when you inactivate this nuclease domain, you move from this to this. You move from a protein that is only able to induce a single strand break instead of two double strand break. And so what's the point? Why would you do that? Because our approach is this one. We have Cas9 nuclease that is with a guide RNA that target the repeat track directly. And so what happens is that you have Cas9 that is going to bind to the repeats. It's going to induce single strand breaks in the expanded repeats. And then with the DNA repair system of the cell, you're going to actually contract the repeats to a non-pathogenic length. And so when we tried this system when we tried this approach in different systems, we found that we actually correct the mutation. We actually contract the repeats. We only contract the expanded allele. We are allele selective. We do not contract the non-expanded one. We have an off-target mutation which is below the detection limit, and as using the nuclease, it's less cytotoxic. And lastly, it's applicable to a multiple disease loci because you're targeting the CAG repeat directly, so the repeat sequence is going to be the same between all of them. And so how did we manage to get to this conclusion? Well, the first thing we've done is trying in the hex cell model that we have in the lab that has GFP mini gene, which is interrupted by CAG repeats, which are present in an intron. And so what happens with this cell line is when you have expansions, the CAG repeats are going to interfere with the splicing, and they're going to be integrated in the mRNA. And so you have non-functional mRNA, and therefore the cells are not expressing GFP, so they are not growing up. But when you have contractions, the CAG are spliced out, and so the mRNA is functional, and then you have cells that express GFP and which are growing up. So that's a really good tool for screening, because using this system, you can see if your drug you're testing is inducing any expansion or contractions. And so when we tried this system in the cell, that's how we look at the data. So here you have the GFP at the bottom and the cell counts, and you have a curve which is the Cas9 with no guide RNA, so it's your negative control. You're not inducing any cuts. And what you do is you do two gates. One gate which is the DMS cells, so the GFP menos, so the cells that has expansion, And the other gaze is 1% with GFP plus, so the brightest cells, so the cells that have contractions. And then you plot your curve with the drug you're testing or your Cas9 case. And if you see that you have more cells in the GFP plus, that means your drug is actually inducing contractions. But if you have more cells in the GFP minus, that means your drug is actually inducing expansions. And so when we tried our system, the Cas9 case, we used it in a Hexel GFP model that has 101 CH repeats. And so on the left, you have the GFP DMS, so the cells that have expansion. And on the right, you have the GFP plus, so the cells that have contractions. And what is that? For the GDM1, which is negative control, you have pretty much like no expansion, a little bit of expansion, but not much. The GCAG guide and the GCG guide, the reason we tried both is because we were wondering, is any of these guides inducing more contraction than the others? We didn't have any clue, so we wanted to try both. And what is you have still a little bit of expansion for both. But what's interesting mainly is that in the GFP plus, so the cells that have contractions, you have much more cells that have contraction when you use the GCAG guide and the CTG guide, which means that your Cas9 case is actually inducing contractions. The second question we had is it time dependent? And the answer is yes. So on the left, you have the same experiment, but on the left, you have 5 days and 12 days of treatment, and you have 5 days of 3 days of treatment for the GFP plus. And what you see there is that you have much more contraction in the 12 days experiments compared to 5 days, which means that you actually do contract over time. The accumulation of contraction is over time. Third question we had was, is it dependent on the size of the repeats? And the answer is yes. So again, same experiment, but this time we tried with 5 different cell lines. So one cell line had more than 270 repeats. Another one had 101, 42, 18, and 0. And what you see there is you have contracting cells in the 270 and 101, but no contraction in the 42, 18, and 0, which means that you actually are allele selective. You only contract the expanded repeats. You do not contract when the repeats are not expanded. So at this moment, we are thinking, okay, we seem to have a proof of concept of Cas9 case that can induce contractions. But as working on Huntington, our main target is going to be brain cells. So we're thinking we need to confirm these results in a more relevant cell type. So we move in Huntington, iPS-derived Huntington astrocyte. And here we've done a small pool PCR. On the left, you have the knee case only. You do not expect any contractions because you don't have the guide. And on the right, you have the knee case plus the guide RNA. And what you have is, you can imagine like a line there, and everything which is going to be above is going to be expansion, and everything which is going to be under is going to be contractions. And when you compare the knee case only versus the knee case plus the guide, you have much more contractions in the knee case treated with the guide compared to the knee case only, which means that you do contract in the HD astrocyte as well. As working with Huntington as well, we're thinking, okay, that's a good thing, but we need to see if we have contraction neurons as well, because the main cells that leads to toxicity for Huntington are medium spiny neurons in the striatum. So we tried in iPS HD neurons, and again, as you can see, we have contraction in the knee case plus the guide, which are much more present compared to the knee case only. So at this point, I'm thinking, okay, we have contraction in HD astrocyte, HD neurons, and we have a guide that targets the CH repeats directly. That means that should work for other genomic loci, right? So we are thinking, let's try another one. So we tried in DM1 neurons, And again, we managed to see contractions for the knee case treated only 9.1% of contraction, and 24% of contraction for the knee case plus the guide, meaning that we do contract a different genomic loci using the same guide RNA and the same knee case. So at this moment, I think, okay, we do have contraction in the Hexel model. We do have contraction in astrocyte and neurons. What about the safety? Because that's one of the main concerns, especially for Cas9. So we looked at off-targets, and we looked at off-target with 6 weeks of treatment with a PCR-free whole genome sequencing. And we did the genome-wide analysis, and we also look at 80 loci with CH repeats, because as targeting the CH repeats directly, these are the most likely to be modified. And we didn't find any mutation on no rearrangement as well, meaning that this approach seems to be safe. The next step after all of this was, of course, in vivo study. And so for this, we decided to move in a R61 Huntington disease mouse model. So we injected the mouse in the striatum, injecting AAVs. at 3.5 months. And then you sequence the number of repeats 1 month and 2 months after injections. And so what on the left is, so you have a pink curve that is the injected mice with the Cas9 plus the guide, and on the black curve are non-injected mice. And from the right to the 0, it's going to be everything which is going to be expansion. And from the left to the 0, it's going to be everything about contractions. And what you see there is that the pink curve is slowly shifting towards the left, meaning that you do have contraction in the injected mice. What you see as well is you see that you seem to lose this bit of expansion here in the injected mice, meaning that, yes, you do have contraction in these cells as well. And if you focus on the contraction index here, which is like only focusing on the contractions, at 1 month and 2 months post-injection with lower values, meaning more contractions, you do have much more contraction in the Nicase plus the guide mice versus the non-injected mice. So we did have our proof of concept at this moment. So in terms of data, so far I've shown you contraction in hex cell model, iPS-derived cells that shows contractions, off-target mutation below the detection limit, contraction in HD mouse. Next question we had was Huntington disease is going to form aggregates of the mutant Huntington protein. So if we contract the CAG repeats, we should technically also reduce the amount of aggregates. So let's have a look. So we had a 2-month-old injected animal, and we did immunofluorescence at 5 months. And so you have a Cas9 only with injection, so in the striatum, and the Cas9 plus the guide. And so I tried to zoom a bit more, but every pink dot is going to be mutant Huntington aggregates. So this one is for the Cas9 only, so you don't have the guides. So you do see aggregates. And when you move with the Cas9 plus the guide CTG, you see that you've lost a lot of aggregates, meaning that you actually do remove also the aggregates for the mutant Huntington protein. And in terms of percentage, we've seen that we have pretty much a 41% decrease of aggregates per cell using this approach. A second question we had was, we've injected mice, we do contract the repeats, we do reduce the amount of aggregates. Do we actually have an improvement for behavior? So we've done a couple of different behavior studies. I'm just going to show you this one for today. This is open field test. So open field test is a good test for measuring anxiety mostly. And so you do put the mouse in a box, and then you look at the time in movement and the total distance that the mouse is going to do. And so on the left here and here, you have wild-type mice that has been injected with either the Cas9 only or the Cas9 plus the guide. And this is mainly to see is if our Cas9 plus guide is going to lead to any cytotoxicity, and the answer is no. And on the right, you have R61 mice that has been injected with either Cas9 Nicase only or Cas9 plus the guide. And what you see there is you do have a rescue for open field distance for the time in movement and the total distance as well. So if I make a quick summary about the data today, we have contraction in a hex cell model. That was our proof of concept showing that we could contract CAG repeats. We have contraction in iPS-derived model with neurons astrocyte and neurons for DM1. We have off-target mutation, which is below the detection limit. We have contraction in the HD mouse model. We have reduction of mutant Huntington aggregates, and we have restoration of some behavior. And so with this, I hope I've convinced you that our approach could permanently correct the mutation. We'll only contract the expanded allele, which I think is really plus when we can be allele selective. We have off-target mutation, which is below detection limits. So hopefully this approach is fully safe. And as using a Nicase, this is what we hope. And finally, and I hope this will be usable for many repeat disorders, because as we've seen, we can contract for DM1 and Huntington disease, but I hope that can be used for a lot of others as well. And so with this, I'd like to thank the Professor Vincent Dion for the lab. I'd like to thank the lab people as well, the funders, and I'd like to thank you for listening to me, and I take your questions.
Do you have a sense of the level of gene expression as well? Is there a possibility that you might be knocking down some of those hits, might be knocking the gene out? And so it could be good because it's knocking down the ones with the long repeats.
Yeah.
But do you have a level, do you know what the level of the mRNA is from the gene?
So I don't think we looked at it. That's one of the things we want to check is we actually think maybe that could be this thing where you just Cas9 bind and then stop the expression. So that's one thing we're going to check, but we don't have the data yet for that.
Hi, great research. So Recently, HT field is also looking at like alternative splicing regulations. I was just wondering if you guys looked at any of those events that have been mentioned in recent papers, and do you think with this method you can see any rescue of those misspliced events?
No, we haven't looked at splice events for Huntington disease yet, no.
The molecular mechanism for your therapeutic intervention, is it related to the putting mutant huntingtin out of the frame due to the contraction?
You mean for the contraction itself?
Yeah, because one can assume that you don't have every 3 repeats contracted. That might be 4.
Yeah. So for the contraction itself, how does it happen? We're not fully certain like which mechanism is fully implicated. We are trying to find out which DNA repair system, which DNA repair protein is implicated directly in it, but we don't have the full answer of the full mechanism, basically. For now, we have mostly like contraction data and molecular behaviors.
Okay, let's thank Antoine.
Thank you.
All right, let's thank all the speakers one more time.